about measuring SNP relevance for Alzheimer's disease.
Yeah. Good afternoon. I know this title is very long, so I will first break it down for you. Anyway, I'm representing the work of a large team, and hopefully I'll do a correct representation of their work. And so let me give you a little bit of background to this work. So we had been working on genomic data for Alzheimer's, and we were using the usual GWAS kind of thing. Most of our colleagues were using that, so we were grinding on this data for a while, and it was a large team, ADSP, so we had multi-universities and multi-organizations, and we took on the task of helping with using also machine learning to that problem, which is traditionally kind of from the space of GWAS and statistics and so on. So it was interesting that the previous talk actually looked at AI methods to a problem that I think traditionally when we were studying it was we were doing mainly using statistical phylogenetics and so on, and recombinations and so on to find out different population structures. So AI is making an introgression into most of the fields, and it is quite interesting actually. So the background of this work is that we wanted to say that we take a space where GWAS and other methods are used. They're giving us some information. Now we will bring in AI or machine learning, and do we learn something more? Can we say something more or different, and how does it compare? And by the time with all this grinding that we are doing over the years, and the reason it took us many years is that every year you have a new release of the data, and the community said, now use the new release of data. So it kind of moved, and by the time we are coming out of it, then there was PRS, polygenic risk score on the horizon. So we said, okay, we'll also bring in the polygenic risk scores, and we'll try to do a comparison here, okay? And so the main idea here is that we want to measure the relevance of the SNPs that you get, okay, which will come mainly from the GWAS kind of studies. So with significance and predictivity or heritability, different communities call it by different names. Heritability or predictivity, that comes from, say, we are calling when you learn something and you predict through machine learning and so on, statistically you're learning, we call that predictivity. For Alzheimer's disease, and so for AI and PRS, we took them two separately, compare AI methods and PRS score methods for Alzheimer's disease. And I took the liberty of adding and COVID, which is not part of the paper because we found some results in COVID data also. So I thought we'll just share quickly that result in COVID. So I guess this unpacking of the title was okay because there are too many components we are bringing in. And so what was the rationale for studying this? So like I said, we started with AD. So Alzheimer's disease is notable for having substantial heritability. That is the predictivity or something that is inherited. But it is unaccounted for by SNP analysis. You only get the strongest signals that you get is ApoE4 alleles, and you don't get much others. And there is a large component of the heritability unexplained. So this was a good playground for us to say that, can we say something more about this unexplained space? So that is the rationale for bringing in all these methods. And so AD presents a unique opportunity to explore what machine learning can reveal about the pathogenic process of the disease, because you have a large heritability here. So how do we do this study? So like I mentioned already while unpacking the title itself, we are going to use multiple methods and trying to get the relevance of SNPs by comparing significance with the predictivity and so on. Yeah, so I'll quickly go over this because I think all of you are very familiar with this. So we use our machine learning methods. We call it RubricOE because, of course, at the core is a machine learning method, but around it, we are running it many times for robustness, resilience, and so on, split test, and that kind of components. I'll go into a bit of this details there. So this just gives you details of what was used and what is the method that you use. Essentially, it's a machine learning method with layers around it just to be confident with what you're getting. And I talked about polygenic risk score in the title. So what we use is we compare it with LD-Pred2. So the first PRS approach, of course, came from Purcell et al. And their labs, but this is the LD-Pred2 is a different method, and we use PRS by using LD-Pred2 method, which is not ours, but which is a popular method that is out there. And again, we use the layers and other precautionary things that we take for that. And this is the data that you can see that we have a detailed data. I think the team was adamant that we do mention what were the data and what were the layers that we did. So this is the June 26, 2021 ADSP data that we are using. It has about 14,000 subjects and about 78 million SNPs that came in as passed by the variant quality score. And the phenotype descriptions includes age, sex, race, ethnicity, and ApoE4. Specifically ApoE4 because it is actually hard to sequence. So in this data set, ApoE4, it had a special focus on actually trying to make sure that ApoE4 alleles are captured, because otherwise they are usually missed by sequencing methods. It's in a difficult, repetitive part of the genome. Yeah, and chromosome 19 contains ApoE and so on, and we also use chromosome 19 and chromosome 11. You know, the way we segmented the data, I was asking him the question, and the first speaker, about subsampling and other things. Here what we did is, when you apply machine learning and you have like 100 million SNPs, what we did is we were breaking them up into chromosomes, the way nature has broken up our whole genome sequence and whole genome, and so we were looking at the chromosomes separately. So like I said, again, Alzheimer's is a challenging problem because it has relatively weak signals for everything except ApoE4. ApoE4, the signal is out of the roof, but for everything else is extremely weak. And you have a general yes, again, this is the details of pre-processing. You can see it later. It is the usual thing that is done in the space, how you take care of maps and other things, and things should be in Hardy-Weinberg equilibrium and so on. So all of these pre-processing steps were done. And yeah, I'm sure you're not able to read it because even I'm not able to read it even from here. So this is the groups that this is the different layers we had on top of the machine learning. Of course, you do split and test because you're trying to learn. And because we are trying to get top features, the method of choice here was SVM. It doesn't give you ordering of the SNPs. To get a rank ordering, you'll see why we needed a rank ordering. We actually split them, and from each split, we try to get aggregate ranking from these methods by using many splits and trying to see to work across these splits to get this ranking. Then we get some rank order of SNPs, and then we go back and apply this machine learning, and then we try to take top 10, top 5, top 15, 5, 10, 15, and so on, and as you know, as you increase the features, its performance improves. But still, you have to make a call as to you have to stop at some points. So that's why we needed the ranking. And even this curve that we plot, which is called the Youden curve, I'll show you, we ran many iterations through it to get this and to decide where to stop the ranking. So let's see if I have, yes, this is, so as you increase the number of features, you can see that the performance is improving, right? It keeps going up. But then you get a little, what we call a bee's knee here. And we see that for the two chromosomes. We tried it on chromosome 11 and 19. And those bee's knees is where we kind of stopped and said, OK, these are the features. Of course, it does go up again. So the performance that you're seeing here, I think it was the Youden index, which is a combination of sensitivity and specificity score. Again, you can use any other score. There's some reason why we picked this. Now I'm forgetting what the exact reason was, but it is taking this into account and doing that. So this was a choice of picking the features. And thankfully, I cut out the part where if you actually look at the mathematical formulation for statistical significance through regression and so on, and you go and look at some machine learning methods, and you look at the formulations, you can come up with a relationship that exists. They're not totally independent, right? You're getting something through significance, you're getting something through learning, but through math, you can actually see they are related. They're not exactly the same. Of course, they're not exactly the same, but there is a relationship. I think in the paper, we probably have the mathematical derivations. I pulled that out from here. I just had the qualitative statement here that the two are related. Well, I think I removed that part as well. So the two we know are mathematically related, but relevance is defined by the relevance of a feature's significance and its contribution to predictivity. That's our definition of relevance. So significance for each of the identified features, both by the AI, which we call RubricOE, and the LD2Pred method is the other method that I'm referring to. That uses logistic regression, and predictivity is measured by the linear SVM that we're seeing. And the order of the presentation is either in LD-Pred2, you use the weights, or in RubricOE was the support. That means how many times you see them in each of the iterations, how many times were you seeing the support, and that gives us an ordering on this. Yeah. So if there is any takeaway, maybe what you want to do is, this is the kind of takeaway that you want to take from this talk, is that you have these red ones is our GWAS kind of p-value. References were also made in the earlier talks. So the red ones give you the p-values, and the green one gives you the support, that is the robustness. In each iteration, if you did 100,000 or 10,000 iterations of it by subsampling and other thing, that's splitting that you are doing, and how many times were you seeing this SNP being shown as your top features, then that is the support. And you see that, and that's the support curve. So it is sort of sorted by the support that you're doing, and then you plot the p-values there. So if you go to chromosome 19, you find that there is a little bit of consonance on this side. That is, you are getting some high p-values there, as well as it is being supported. So you would have kind of more confidence in that. This is a little bit, you'll have to take it with a grain of salt, or you have to do a little more digging in to see that why you are getting some p-values, high p-values here, but you're not getting enough support for those guys. So this is for RubricOE, and I will quickly go to the result from LD-Pred2. And LD-Pred2, this is my interpretation, and this is our interpretation, is that again, the figure is the same. You are comparing the so-called p-value with the support score. Now the support score is coming from LD-Pred2 here. Here it is not clearer than as it was in RubricOE, because you did see a high value and then it was falling down. So of course it's subject to interpretation, but this is the result that you get for. So that means you're taking p-values and comparing it with the robustness or the support coming from the method. And of course, we had said that in the beginning, one of the points is, can machine learning learn anything new about it? So we want to see that, can we make any biological connections? Similar to the first speaker, so we did an analysis. We take the SNPs that we get and go and look at what are the biological pathways that they're involved in. So these are the top 3 pathways that we pulled out, did the usual calculations and so on. So machine learning is giving you some way of trying to also make some biological connections with the disease itself, because your phenotype is disease and non-disease. So that's your phenotype. So this is the connection that one is making. And like I said, we also did the same approach on a different disease set, which is COVID from the UK Biobank data set. And we do a similar procedure. And of course, there is a little bit difference in the architecture of the pipeline here because of the nature of the data. Probably not important, but we wanted to show that slight difference in architecture because we are including EHR here, along with the phenotypic data that we had earlier, which was like age, sex, and so on, but we have detailed EHR here. And another thing about this work, which is not part of this work, but I took the liberty, the poetic license I get as a speaker of inserting some other work as well. Here was that severe COVID something is not a phenotype in UK Biobank data. It doesn't exist. So this is some inferencing we did from where they're hospitalized, and let me just see, that might just be, yeah. So severe, we had to define the severe cases where they hospitalized positive samples, and you actually remove the hospital-acquired COVID-19 because the frontline staff probably were getting tested very often, and if they got it from the hospital, they were not taken as severe COVID. So we had to kind of infer from the phenotypes what are possibly severe COVID cases. And based on that phenotype, we did this. And here, again with COVID, we did a similar exercise of going and figuring out biological significance. And it turns out when you do that, you get two pathways that you expect to see in them, and then you also get some new pathways that are perhaps correct, but that have not been reported elsewhere. So it is a hypothesis that there are certain other pathways that may have played a role in severe COVID. So this is my last slide on the conclusions. So machine learning, any machine learning method, such as our method called RubricOE, tends to present more candidates than GWAS significance-based approaches, but they yield similar predictive power. The second very subtle point here is that SNPs with low significance do not tend to boost predictive power, but also do not tend to hurt predictivity power. So they are compatible in that sense. And so with sufficient signal, both approaches will show some consonance with significance, but this tends to be lost when the signal is too weak or low. But that gives us gives us a way of trying to make some biological connections. I said that was the last slide. Maybe I have one more. I'll just stop with that. So we have this. We have this on GitHub, and this is accessible, called Geno4SD, for the different pieces of algorithms that I talked about here, and there are some more. If you're interested, you can go and try it out. I don't know if this, okay. I will stop there, and thank you for your attention.
Time for a couple of questions.
Thank you. Maybe I don't understand, but because you say this is essentially machine learning, and there are some buzzwords there like AI, machine learning, even COVID, but what exactly? Like, is it a linear SVM?
Yes, linear SVD,
Okay.
a support vector. Yeah.
Okay, but that is what is this RubricOE is?
Yes. So RubricOE, maybe I moved too fast. It was not clear. SVD is what we are using at the core, linear methods, linear kernels. And it has other layers of splitting and the robustness calculations that it does, the Youden curve and so on. So it has those other layers built in, so you don't have to do everything else. But at the core is this machine learning method.
So it's, but maybe this is my comment, but maybe it's not the conclusion that you cannot then generalize this to all machine learning, right? This is just one approach.
Yes. Okay, so that's a good question. So why did we take only this method, right? So when we tried, that's why it was called rubric and so on. So we tried a whole bunch of machine learning and AI methods, okay? And it used to automatically try to tell us which one is the best one. And time again, it was this method that did the best. So we said, okay, let's not waste our time with the other method. Let's go with this. But you're right. But there was a reason why we went with this method. It was consistently doing well, at least in this kind of data set. Not able to make an extremely general statement, but yeah, that is what it was.
So this finding that on different chromosomes, you see this concordance between p-values and these Youden curves you get from the
Stability is core.
stability curve.
Yeah.
So is that due to the kind of the LD structure across these different chromosomes, or what do you think?
Right. So no, LD structure, I would rule it out because in our pre-processing and other methods, if they are related, we just take one representative. So in a sense that the representative in the plot that you would see, it is not there with its LD companions, approximately. So we do remove that. The consonance that we are seeing is perhaps because it's a true signal. Assume that there was a true signal, something that really works, then it is going to have high p-value, and it will be you will keep seeing it because it will be stable, and no matter what you change, you keep seeing that as a result. So our way of saying that it is perhaps a true signal is because we are seeing a consonance through completely different ways of looking at it.
Yeah, so what are the characteristics of the SNPs that the rubric method found but GWAS wasn't able to find? Were they like rare variants that maybe GWAS didn't have enough power to detect, or?
Right, good question. So in the pre-processing that I just rushed through, extremely rare variants were removed. Okay, so if they're extremely rare, we didn't take. So it's a good question. So we focused on what we found in both sides, but we didn't go and look at what this found, but this didn't, or this found and this didn't. So that was not something. So our assumption was that it's possibly noise, or possibly it's not standing the test. So we didn't actually go and examine closely because we were looking at consonants from these two sides. And the biological pathways that we showed were coming from the intersection, or what both the methods were saying works. So stability and p-values.
And also, just to clarify, so the method does not take any additional information other than the GWAS.
Yeah, does not take any.
Okay, so just like a post-processing on top of the.
No, so sorry, could you repeat the question again?
For the rubrics.
RubricOE, so here is the input you have. You have the SNPs and the phenotype data. GWAS will go and do its stuff. Okay, ridge regression, GWAS will kind of go and do its stuff, and then machine learning will do its stuff on the same data. If you have filtered out the SNPs in the pre-processing method, that's fine. So you will just take the SNPs and give it to both methods. So one is not feeding on the other; they're working independently on that.